So a very warm welcome to the EU Pearl webinar on non-concurrent controls. It's a great pleasure to see the great interest in this topic and how many people have registered and joined today. My name is Martin Bosch from the Medical University of Vienna, and I will co-moderate the event together with Peter Masonbrink from Novartis. Next slide. As people are still joining, let's start with some housekeeping rules. Please note that the session will be recorded and made publicly available. Only the panelists and speakers can speak. All other participants are muted. However, you can ask questions at any time using the Q&A tool in Zoom, and we and all panelists will try to answer your questions immediately there. To make the webinar even more interactive, we also have prepared some polls with specific questions on non-concurrent controls. Next slide. To set the scene, the choice of control groups is crucial for the generation of robust evidence on the safety and efficacy of novel treatments in clinical trials. And the accepted gold standard are the concurrent control groups in double-blind randomized trials. However, there are difficult experimental settings where also other types of controls are considered and intensively discussed, ranging from control groups of historic randomized trials to cohorts drawn from real-world data. The non-concurrent controls within the same platform trial are a new type of control group, which are collected under the same overarching protocol and standardization. In this webinar, we will learn about methods to use these controls, and we will discuss to which extent these methods can adjust for potential biases when using such non-concurrent controls. We will hear the perspectives of different stakeholders if and in which settings they can be used. Next slide. So the agenda for the next 3 hours is quite busy. We will start out with 2 presentations on frequentist and Bayesian and statistical approaches to incorporate non-concurrent controls in platform trials. Then we will hear about the relevant current regulatory guidance on this topic. In the second part of the webinar, we hear academic, industry, and regulatory perspectives and experiences on the use of non-concurrent controls. In the final part, we will have a panel discussion based on several pre-agreed questions. I said, please raise additional questions at any time using the Q&A tool in Zoom, and the speakers and panelists will answer them during the meeting. We are very glad to have such a distinguished set of experts and key opinion leaders with regulatory, academic, and industry background in the panel, together with several contributors from EU Pearl. Next slide. Before we start with the topic on non-concurrent controls, first let me shortly introduce EU Pearl. EU Pearl is an EU-funded project by IMI, the Innovative Medicines Initiative, to form a strategic alliance between the public and private sector to transform the way clinical trials are conducted. The aim is to develop a common framework for platform trials with the objective to improve the drug development process. The approach is patient-centered, and the framework is tested in 4 disease areas, from more prevalent indications as depression up to rare diseases like neurofibromatosis. This common framework shall enable a structured discussion to design platform trials in different indications, considering when and how to incorporate non-concurrent controls. Next slides. So let's start with the methodological presentations. The first speaker from EU Pearl is Marta Bofill-Roy. She's a statistician and postdoc at the Medical University of Vienna and will present on the frequentist approaches to incorporate non-concurrent controls. Marta, the floor is yours.
Thanks a lot, Martin. Hi, everyone. Next slide, please. Okay, hi everyone, and thanks for the introduction, Martin, as I said. Today I'm going to talk about model-based adjustments to incorporate non-concurrent control data. This work is part of the EUPIL project, and I would like to first thank to the members of the working group of non-concurrent controls. So the work that I will present today is based on this paper available on arXiv. Next slide, please. So I will start this presentation with a short introduction. So platform trials are multi-arm, multi-stage trials with the extra feature of allowing experimental arms to enter and leave the trial at different times. The number of treatment arms are not predefined, and the common infrastructure allows for the possibility of sharing a control arm. Here we can see an example. This trial starts with one experimental arm, and later on a new treatment is added in the platform, and after that 2 more treatment arms are added, and all of them are compared to a shared control arm. Next slide, please. So sharing control arms in platform trials leads to 2 different types of control data. Concurrent controls are the group of patients recruited to the control when the experimental treatment is part of the platform. And non-concurrent controls are patients recruited before the treatment and entered the platform. So as an example, here there is a trial with one initial treatment and a second one that enters later. So for the second treatment, the non-concurrent controls are the group of patients that are part of the trial when this arm is in the trial, so in dark gray, whereas the non-concurrent controls are those that were in the platform before, in light gray. Next slide, please. If another third arm is added, then the concurrent and non-concurrent controls will be different from the previous ones. And since this arm started later, then there is a larger number of non-concurrent controls patients. Next slide, please. In randomized control trials, we use randomization to avoid potential biases due to external factors and to come up with comparable groups. Non-concurrent controls have been randomized too, but in different sets of treatments that are changing over time and whose patients were randomized at different calendar times. Incorporating non-concurrent controls can improve the efficiency of the trial, but the lack of true randomization over time might introduce bias due to time trend. So the question that arises is whether we can use non-concurrent controls and how can we do that in order to increase the efficiency of the trial while inferring valid conclusions. So in terms of the analysis, the standard approach is the so-called separate approach, which only uses concurrent control. In the opposite side, we can consider the pooled approach, which consists of using all the controls all together without any adjustment. What I'm going to present today is the frequentist model-based approaches, which adjust for time trends by including time as a covariate. However, at the end of the presentation, I will mention other approaches as well. Next slide. So let's assume we want to compare arm 2 versus control, and arm 2 enters when the trial already started. We define period 1 as the time interval in which the arm 2 was not present in the trial, and period 2, the time interval in which the arm 2 is in the platform. And the goal is to test the effect on treatment 2 against control, here denoted by theta. Next slide. So we consider a regression model for the expectation of the response, here assumed continuous. So to fit this model, we take into account all the treatment arms to adjust for the time trends. So in this example, this means to use as well the data from arm 1. The first covariate of this model is the baseline, and then there are the covariates referring to the treatment effect for each arm, and then there is an extra covariate referring to the time period effect. And this time period effect is fitted with the control group data and the data from all treatment arms. Next slide, please. So how the estimator of the treatment effect looks like under this approach. Let's consider the sample means per arm and per period, so that the first subindex corresponds to the arm and the second to the period. And let's define rho as a parameter that weights the sample size of the concurrent controls over the sample size of the concurrent and non-concurrent controls and arm 1 data. So the treatment effect estimator when using the separate analysis is just the difference between the sample means of concurrent controls and arm 2 data. Next slide, please. When using the model-based approach, this estimator is the difference between the sample of arm 2 data and an estimator of the control that accounts for concurrent and non-concurrent control data. Indeed, this estimator is a weighted mean between the concurrent control mean and the non-concurrent control mean, adjusted by the time trends by means of the difference in response from period 1 to period 2 in arm 1, as we can see in the last equation in this slide. Next slide, please. The reduction in variance when using this estimate for the control over the estimate used in the separate approach is equal to the weight rho. So the reduction will increase with the non-concurrent control size, and on the other hand, if the concurrent control data increase, no reduction will take place. Next slide. From here, we can see, or we can show that the treatment effect estimator of the model-based approach is unbiased as long as the treatment time trends across arms are equal and additive in the model scale, no matter, however, the functional form of the time trends. Next slide. The model-based approaches, the model-based approach rely on the model assumption about the functional form of the time trends, which is assumed to be stepwise. Also, when using this model, we implicitly assume that the effect of time is equal across arms and additive in the model scale. Next slide. In our work, we evaluate the robustness of the corresponding hypothesis test with respect to the these assumptions, and evaluate under which circumstances they lead to a gain in power and a type 1 error control. Today, I will illustrate that through a simulation study. So as I explained before, I will focus on testing the effect on arm 2 in trials with 2 treatment arms and the shared control. And what we did is we simulated trials with continuous data in the presence of linear and stepwise time trends. To simulate linear time trends, we considered patients indexed by entry order in the trial, and that the time trend linearly increases with time. So we can see here as an example how the means increase with the recruitment. And the strength of the time trend is given by the slope of this line, which we denote by lambda. When simulating stepwise time trends, the change in the time trend occurs when we move from period 1 to period 2, and the strength of the time trend is given by the jump from one period to the other. Next slide. So we start investigating the power in trials with equal time trends across arms. Therefore, we consider that there is an effect on arm 2 and that the strength of the time trend, that is to say the parameter lambda, is equal for all arms. Also, we consider that the patients were randomized using block randomization. In the following figures, we illustrate the power of testing arm 2 versus control depending on the pattern of the time trend and according to the strength of the time trend, which we vary. So what we are doing is that we are considering different values for these slopes and jumps that I was talking about before. So in this plot, in green, we plot the power when using the separate approach, that is to say only using concurrent controls. In blue, there is the results using the pooled analysis, so namely pooling concurrent and non-concurrent controls without any adjustments. And in red, we plot the power using the model-based approach. So from this plot, we see that incorporating non-concurrent controls by means of the modeled approach improves the power as compared to the separate approach. Next slide. Now we assess the type 1 error rate in trials with equal time trends across arms. We simulated then under the null hypothesis, and the strength of the time trend is assumed to be equal in all arms as before. So we plot the type 1 error according to the strength of the time trend and depending on the pattern of the time trend. And what we see is that the separate approach maintains the type 1 error and that the pooled analysis leads to an inflation for positive time trends. The model-based approach performs similarly to the separate approach so that both the separate approach and regression model curves overlap, and thus the model approach maintains the type 1 error despite the pattern of the time trend. Indeed, in the paper, we show that this holds asymptotically for both binary and continuous endpoints and for different time trends patterns other than linear time trends. Next slide, please. However, what happens when we have different time trends? Let's now consider that we have same time trends for treatment 2 and control arms and different time trend for treatment arm 1. So unlike the previous case, here the strength of the time trend might vary between arms. We plot here the type 1 error with respect to the strength of the time trend in arm 1, here denoted by lambda 1. And what we can observe is that when we have equal time trends for the 3 arms, that this happens at 0.1, then the model-based approach maintains the type 1 error. But when there are different time trends, then the type 1 error control is lost. Although it's not shown here, the power using the model-based approach is sensitive to non-equal time trends and might even be reduced in some circumstances. Next slide. But are these cases plausible in real life? So different time trends across arms happen when treatments have an effect over the control, but external factors alter the response to arms differently. Examples of these might be vaccine trials where different variants of the virus are predominant over time and the vaccine efficacy depends on the variant. Also, time trends might be additive in the scale of the model. So for example, for binary outcomes, the time trends can be equal on different scales, such as the risk ratio instead of odds ratio scale. However, even if there is a risk of bias, the reduction in variance might outweigh the potential bias introduced when using non-concurrent controls. But in any case, a case-by-case assessment should be considered to evaluate the plausibility of the assumptions and the potential risks. Next slide. So to sum up, the model-based approaches, or this model-based approach controls the type 1 error and improves the power under the assumption of equal time trends in all treatment groups and correct the specification of the time trend scale. The model leads to unbiased estimators, even under misspecification of the time trend pattern. Moreover, this model results in unbiased hypothesis tests if block randomization is used. And in this presentation, while we have considered only a simple trial design, this model can be generalized to trials with more treatments and more periods. Next slide. So as I said, I've been focused so far on rather simple models, but alternative models can be considered. For example, we can use models adjusting for different time trends per arms, but the gain in power in this case will not be large, as the non-concurrent control data will not contribute to the effect the treatment estimator. We can also consider smooth estimators for the time trends, such as splines, but this flexibility might come with less power. Bayesian models can be considered as well, in which the time trend parameter smoothly estimates the effect of time, borrowing the information from one period to the next, as Scott will explain now. However, using these other modeling approaches rely on the equal time trends assumptions as well. Next slide. If instead of modeling, we want to downweight the non-concurrent controls, we might consider approaches proposed to incorporate historical controls, such as the test and pool, the weighted pooling approach, and map approaches. In the test and pool, we first perform a hypothesis test to compare the response rates in the concurrent and non-concurrent controls. And if there is a significant difference, then a separate analysis is performed. And if it's not, we do a pooled analysis. The weighted pooling is based on estimating the response of the controls by weighting the information from concurrent and non-concurrent controls using a pre-specified parameter. In the map approaches, we define the prior for the control using a mixture of two priors. One is the prior of the concurrent controls and the other is the prior based on non-concurrent control data. And this mixture is weighted by a parameter that represents the degree of borrowing. So when using all these models, the validity of the results might strongly depend on the pre-specified parameters and assumptions we made. Besides, none of these approaches control the type of error in all the scenarios. Next slide. So in this presentation, I focused on the bias triggered by time trends, but there might be other sources of bias when using non-concurrent controls. In particular, a statistical inference relies on the specification of the analysis methods before the data becomes available. However, in platform trials, the non-concurrent control data might be published or reported when arms are completed. And as a consequence of that, the addition of new arms to the platform might be biased due to previous knowledge on how the controls are so far in the platform. Therefore, strategies to mitigate these biases should also be considered. Next slide. So wrapping up, non-concurrent controls might improve the efficiency of the trials, but also they can lead to bias if we do not adjust for time trends. We proposed a model-based approach to incorporate the non-concurrent controls, but if using this model-based adjustment, then we need to bear in mind the assumptions being made. In particular, the validity of the results will depend on having equal time trends across groups and the correct specification of the scale of the time trend. Thanks a lot.
Yeah, thanks a lot, Marta. Our next speaker is Scott Berry from Berry Consulting, which is well known for the design of innovative Bayesian clinical trials. Scott, please go ahead.
Thanks, Martin, and thanks, Marta. That was very good. I'm also a part of EU-PEARL. I'm going to talk about, as Marta described, a Bayesian approach to this. I'll describe a couple examples of this. This is joint work with a number of people at Berry Consultants, and we've submitted a paper that has many of these results. Next slide. So here's an example. This is public on the iSpy2 website. This shows the multiple treatment arms since 2010 that have joined the iSpy2 platform trial. You can see the time in which these treatments were initiated over the course of the trial. This provides an example of the sort of overlapping active arms in the trial with the relative control arm. Now, I'll ignore the fact within some subsets of these of breast cancer, the control has changed, but I'll ignore that for this presentation. Next slide. There's a general sort of demonstrating that non-concurrent controls are this separate, disjoint set of historical data that might be from a similar population or not. This shows within a platform trial, the non-concurrent control. So this shows an example of a particular active treatment arm where we do have concurrent controls to that active arm. There were patients enrolled previously to the control. These are not separate, disjoint, disconnected assignments. There are multiple active arms that all overlap over the course of the trial. So it's a connected network, if you will, to the back, all the way back to 2010 for these particular patients. By the way, this demonstrates my initial interest in this question was one of comparing athletes of different eras. Could you compare Tiger Woods, Rory McIlroy to Jack Nicklaus, Tom Watson, Ben Hogan? They never played at the same time, but they played with athletes that did overlap. You have this incredible connected network, and it's really very much the same problem. It's actually a little bit harder because athletes age and over time differently than treatment arms age over time. So the question is what to do with these non-concurrent controls. Next slide. The there's a sort of pejorative thing. Calling something historical is pejorative to many people. These are very different, as Martin pointed out. They're enrolled in the same platform trial. I'm talking about, in this example, non-concurrent controls that would be eligible for the particular arm. So we know the inclusion-exclusion criteria. They have the same data quality, the same visits. They've been randomized. Everything about these patients are the same except for time. There's a covariate that differs in these patients, but of course it's a randomized trial where we have this covariate recorded. We can adjust for it, which is what Marta just described. So next slide. Here's an example brought on by the iSpy 2 trial, but many trials, where we might be interested in arm 5, the pink arm at the end here against control. Control is black, shown from beginning of time 1 to time 10. I have created different eras, seasons, or parts to this trial, the 10 different eras of this, in which the last 4 eras, arm 5 and the control arm, have overlapped. The control arm was before this with other active arms you can see in the other different colors. I don't attack this problem from how do I control type 1 error, what's my bias. My question is, how do I model this to make the best inferences to compare arm 5 to control? So next slide. A very common way to think about this is, let's think about this as 10 different trials. We have 10 randomized trials that, let's assume the randomization is fixed here. Each one of these has fixed randomization. They just have different active arms. The last 4 of them have control and pink. The previous 6 do not. They have other arms. They have control, but other arms. And of course, those arms overlap pink and overlap arms that overlap pink. So how would we model this? It's very similar to a meta-analysis. It's not a meta-analysis. It's all one trial, but it's meta-analytic models. So next slide. Very commonly, we would model this a log odds model, where we model alpha as the study effect, which is really a time effect in the model. And then we have an effect for each treatment. I'll call it beta for color for each particular treatment in this setting. Very common model that we would use. The question is, how do we model the alpha? Marta described an example where you model these with fixed effects. If you click the next slide, I think it brings a bullet. We rarely in meta-analysis would you model these as fixed effects. Stein has shown us that we can do better than that. We typically would use hierarchical random effects models for study. The difference here is that we know these are not exchangeable. 2 and 9 might not be exchangeable. They're ordered. They're a time series. So we may very well do better by recognizing the order of these trials in time, that 5 is much more likely to be closer to 4 and 6 than it is to 10. So if we move forward, we can model the alphas, the study effects or the time effects, next slide, with a smoothing spline. So you can model the alphas with some way of time, and we would not do this with a linear effect. We would not do it with even a monotonic effect. So we use a smoothing spline, a Bayesian smoothing spline to model time, where we can learn, by the way, if there's a time effect, and if it appears there's not effect, or it's linear or it's flat, you learn very quickly and there's smoothing of that. So this model, this Bayesian time machine, is modeling the underlying effect of time with a Bayesian smoothing spline. Otherwise, it's very similar to what Marta presented with the modeling of time. So if we move forward, I'll show you a quick example of this. In this exact example, I'll compare 4 different approaches. The first, of course, is you can just model the concurrent controls, ignoring an incredible amount of data by just restricting to that. This shows an example, a frequentist logistic regression analysis of the concurrent controls. If you click the next slide, you could pool them. And this is typically this sort of pejorative term to using historical controls. It depends how you use them. It's critically important. Is if you just pool them, we recognize that that creates bias. It doesn't adjust for time trends, and you can see the example of modeling it this way, which would be a bad way to analyze these data. And we know it's a bad way because of the overlapping arms. The next slide. This is modeling them as fixed effects. So you don't model the alphas with any sort of model. They're all fixed effects. And you can see the analysis of that. Notice the standard error here. The log odds ratio is going to be estimated very similarly, other than 2, which is a bit of a straw man here. You'll see the standard error approach. And then the fourth one here, if you click next slide, is the Bayesian smoothing spline. You can see the underlying estimate of the effective time has been smoothed across it using all the arms, not just these 2 arms. It's using all 6 arms in the trial. And it gives the estimate of pink relative to black using the smoothing across the entire range of the platform trial. Notice the standard error has decreased in this a little bit with relatively similar estimates of the treatment effect. We give a Bayesian posterior probability of superiority for the Bayesian smoothing spline here. So next slide, Marta set it up sort of the simulation of these approaches. So here's 3 different assumptions, and I am assuming additivity of time across these different simulations with different shapes of time. If we go to the next slide. I'll highlight this in the relatively short time here of comparing the different approaches. First of all, the bias of this. The time categorical, where you model time as a fixed effects, is going to be unbiased under the assumption of additive effects, as is concurrent controls. The smoothing, you can get small biases with the smoothing. It's essentially unbiased. In our model, we use this model in a half dozen to a dozen platform trials. We pick small buckets of time. We don't pick them to be when arms come and go in the trial. We might do it every month is a time unit, every 2 weeks is a time unit, or every quarter is a time unit throughout the trial. So we're picking relatively small buckets and smoothing within those circumstances. And that's going to enable us to get unbiased estimates in the Bayesian smoothing. The time machine is the 4th one here. On the right, I've shown the mean squared error relative to the time machine. And the time categorical has, across these scenarios, a 6 to 8% increase in mean squared error relative to the time machine. It does pretty well. The concurrent controls in this example for arm 5 has about a 20% increase in mean squared error in that scenario. Hence, the ignoring of data and the inefficiency of restricting your analysis to a subset of data by taking concurrent controls. So if we go next slide. So it's recognizing that we have many overlapping arms in these platform trials. They come and go at different times, demonstrating in a number of scenarios the cost inferentially of ignoring that data and not modeling time within that. Fixed effect modeling, we do that in trials and it works. I prefer the smoothing. We understand something about time, the ordered aspect of time. The really important thing to point out here is this is not running a single trial where we suffer a 20% increase in mean squared error for the single trial, which maybe we can overcome with a little bit of sample size. In that circumstance, this is a perpetual platform trial. In I-SPY 2, there have been 25 arms. This is multiplied across all of these arms. It's 20% for every arm, and that can even increase as we get more data. So you're just continually costing yourself inferential power for all of the arms in the trial. Every arm increases in size if you restrict it to concurrent controls, which might mean you investigate less arms. You take less shots on goal. It has huge ramifications across a perpetual platform trial. It's not a one-shot trial where we can overcome it. There are operational and data sharing aspects of this. In order for Arm 5 to do their primary analysis, you don't just need the non-concurrent controls, you need all the other active arms in order to estimate time. So the primary analysis needs all the arms. So this is a new aspect of data sharing, of being able to do this primary analysis. So this is a hurdle in some cases, but one well worth accomplishing and using within platform trials, and recognizing this is an important aspect of it. All right, I will stop there. Thank you.
Thanks, Scott, for the great talk. And we now come to the next presentation, which is given jointly by Katharina Hees from the German Regulatory Authority, the Paul-Ehrlich Institute, and Ursula Garczarek from CITEL. Katharina and Ursula.
Thank you, Martin. So Ursula and we will present some results of a review of regulatory guidelines, which work was part of a LASA research we conducted within the working group of non-concurrent controls in EUPERL. So, and I have to present the standard disclaimer here. So the views which we are presenting here are our own. Next slide, please. So when we started this research, we started with looking for platform trial related guidelines and guidelines which were saying something about non-concurrent controls in the strict sense. But there are so far not many guidelines available that address this issue. More precisely, we have found exactly these 4 here. So there is the very recently published FDA guidelines on platform trials in the COVID-19 context. And there's also the FDA guideline on complex clinical trials, as well as the overarching master protocols FDA guideline. So the only available European document is the CGFG recommendation paper on the initiation and conduct of complex clinical trials, which was published in February 2019. So all in all 4 documents are very vague regarding the recommendations for the use of non-concurrent controls. So more or less, they are all raising the issue of a potential time drift. And due to this, the FDA guideline for master protocols, the one here on the top, is relatively clear against the use of non-concurrent controls. Next click. So far, no instructive guidance is available regarding the use of non-concurrent controls, but then the story would already be over here. So next slide, please. We have extended our review to guidance provided regarding the use of external or historical controls in clinical trials, since non-concurrent controls are in the end just a special case of external controls. We will later also discuss in what points they are maybe better. So we downloaded all guidelines from the EMA, ICH, FDA, and screened them for the printed keywords. That means we also look for the keywords external or historical borrowing and so on. And after some pre-screening, for example, we excluded guidelines which were related to medical devices because they are not part of EUPERL. Or we deleted guidelines which were using historic controls just in a preclinical setting or for the planning of a trial. And after this pre-screening, we ended up finally with 37 guidelines. And for all these 37 guidelines, we filled out a before pre-specified questionnaire. And this was filled out for every guideline by 2 reviewers. Next slide, please. Okay, so very short, on this slide, I wanted to present you the age of the guidelines we have identified. It's very obvious that most of the guidelines are quite new. So next click, please. There are also some guidelines which are a little bit older, I would say. They are all rather general and established guideline. And what's maybe also important here is to mention the ICH E10 guideline, very famous guideline on the choice of control group, which I would say is maybe the most informative guidelines concerning the topics of external controls. Next slide, please. So one of the questions of our questionnaire was, in case that the guideline deemed the use of controls as acceptable, then we looked into these guidelines and asked what are the specific circumstances mentioned in which the use might be acceptable. And these are here what we found in these guidelines. So the most often mentioned circumstance was a rare disease, or also a large treatment effect, and ethical concern, unmet medical need, predictable disease course, high mortality, a pediatric trial. In this context, this was just more in the sense of extrapolation from adults to the pediatric patients. What was also quite often mentioned was that there should be no time trend, that would be good to have an objective endpoint, or also a circumstance in which it might be acceptable that the randomized trial is not feasible. Next click, please. So it's the question, which of these are relevant if we are thinking on non-concurrent controls? I would say some of the points are transferable, maybe not all. So I would say it's maybe, for example, a large treatment effect or no time trend in disease course or objective endpoint could all be circumstances in which the use of non-concurrent controls might be more appropriate if it would not be such a circumstance. Okay, so next slide, please. Another question was in the questionnaire, which method are mentioned in the guidelines? So the classical methods can be divided, I would say, in Bayesian methods. Most famous here are probably the map prior with the one which Marta already mentioned in her presentation, or also the power prior approach. Another possibility are modeling approaches. More precisely, we are referring here to classical regression models, such a method as Marta was using in her research. Another possibility are, of course, also matching approaches, like, for example, a propensity score matching, or you could also do a classical meta-analysis. A more rudimentary method is the one on the top, so to just use the historical controls to define a threshold for a hypothesis test. So overall, one can say, therefore, next click, please. So the guidelines remain very vague rather than instructive on methods. So they were just saying one sentence, so you can use a Bayesian approach or can do a matching, but they were not really saying a lot concerning this. Next click, please. So concerning the relevance of to non-concurrent controls, I would say most of the methods can also be used for the analysis of non-concurrent controls, maybe for some, for example, to model the time trend. The situation, as Scott already pointed out, is in such a platform trial maybe a little bit better because you have this overlapping time periods. Okay, saying this, I would hand over to Ursula. And next slide, please.
Katharina. So when we looked into the concerns that were raised in the guidelines, we found to no surprise that comparability and bias are the main general ones that are mentioned. There were also indication-specific and other concerns to which I will say something later. The more specific sources of non-comparability and bias below are interesting to be looked at to compare the risk for these between external controls and non-concurrent controls. Note that this is an interpretation of our own and not directly derived from the guidelines. Next slide, please. So heavy selection bias for external controls can come through willful bias by cherry picking or through mechanisms like publication bias in the availability of data. Both of these high risks are much reduced for non-concurrent controls. Strong risk of differences in measurements for external controls come through different standard operating procedures or instruments that are used to determine the endpoints. In platform trials, that can be prevented by careful planning. Yet, shifts and drifts in instruments for newly developed methods or operator bias still need to be attended in platform trials. Next, oh no, data, big risk for data and trial integrity arise with external controls, especially in single-arm trials when they are combined with single-arm trials where there is no masking. That would not be relevant for non-concurrent controls whenever in that platform trial masking is possible. Though integrity issues very comparable to any adaptive clinical trial arise in platform trials, whenever interim results become available, the behavior of investigators and patients may change. The risks for changes in standard of care are a bit like the differences in measurements. They are under the control of those who perform the platform trial. Whenever a shift or a change in the standard of care is necessary, one can then make a very informed decision on whether or not to use the controls that are affected. Next slide, please. So when we looked into the requirements, they are echoing what we have already said with the concerns. So the population must be comparable, and the data needs to be of high quality. And so traceability and standardization are quite important aspects for that. Next slide, please. And for non-concurrent controls, especially the quality of the data is fully in control of those who plan the platform trial. And as said, with the comparable population, we are in a bit better position as compared to external controls, but there are still things that really have to be taken care of. Next slide, please. As said, some words on indication-specific mentions in the guidelines. Most of what you see in this slide is actually more interesting to those people that want really to use external controls in their trial. What we want to highlight with this slide or these findings is that there are very instructive guidelines on trial designs with external controls that address specific questions under very specific circumstances. Next slide, please. And the same holds true for the concerns. There are also very clear instructions out when not to use external control in other specific situations, which may mean for non-concurrent controls that fundamental disputes about the right choice for the variance bias trade-off one has to make may be settled when we are looking into specific platform trials for specific indications and drug types. That's the moment I want to hand over to Katharina again. Next slide, please.
Thank you, Ursula. So I will close with a summary of our key findings. So I would say overall, one can say the guideline availables are rather vague regarding the use of non-concurrent controls, at least in the strict sense. So in this talk, we interpreted findings from external or historical controls for non-concurrent controls. So a lot of issues that decreases data quality for sure no longer exist in the context of NCCs compared to external controls. Comparability is also better, but I would say it remains still an issue. So the concerns about selection bias and changes in measurement are reduced, but the concerns about change in standard of care and time trends are still an issue. And I would say a good example is here probably the FDA guideline for master protocols in COVID-19. So the concerns about data and trial integrity are probably similar than in the context of an adaptive clinical trial. So one other key finding for us was that looking at the indication-specific guidelines has strengthened our conviction that they have the highest potential to be instructive. Since for us, the big question, what is over this webinar to use or not to use, strongly depends on the specific indication. So thank you very much for your attention.
Yeah, thanks a lot, Katharina and Ursula, for this comprehensive overview on what the regulatory guidance documents says on non-concurrent controls. We see that there is yet little specific guidance on non-concurrent controls and platform trials. However, we have several regulators on the panel and look forward to hear later on their perspective. And with that, I hand over to Peter to introduce the stakeholder representatives.
Thank you, Mark. We will have a perspective given by industry, regulators, payers, as well as academia, and the first presenter will be Olivier Collignon from he will give industry perspective.
Hello, good afternoon, everyone. Very happy to be here. Thank you as well to all my colleagues on the pharmaceutical industry to have helped me preparing those slides. And maybe while the slides are loading, maybe we could go to the previous slide. So okay, well that's all right. Let's start. We remove the slide with the names of the participant. That doesn't matter. Just wanted to thank my colleagues again from the pharmaceutical industry who helped me preparing those slides. And really, one of the points that came up is that is there really such a thing as an industry perspective on the topic? I think when we started talking on that topic is that we're driven a little bit by our experience, but also mainly by the state of the art. And a lot of information can be found in the literature. So can we go in to the next slide, please? Thank you. So there's a whole set of very clever people who have started to work on the topic of non-concurrent controls and platform trial and how and when should we add them. You have them on this slide on the left-hand side, and I would like particularly to bring your attention to the last one, which has been written by the American Statistical Association. We really provide an excellent review of the different views on the matter. And you could argue that the principal components really of these views are summarized here. So in the absence of time trend, we have a potential gain in power and precision, has been demonstrated at the beginning of the workshop. When we have a time trend that is not accounted for, then we are prone to estimation bias and type error inflation, which of course can be an issue in a confirmatory setting. The good news is that a time trend can be modeled for and accounted for, and under the right assumptions, we can maintain the power gain while controlling the type 1 error rate. But the bad news is that we never know the true model. And that's the issue, specifically when we don't have the data yet. For example, when we need to write the statistical analysis plan. So in an area where you need to control the type 1 error rate, it's probably safer to restrict the comparison to concurrent controls for confirmatory objectives, for example. And this is where having different estimates and different sensitivity analysis will come into play. For example, with and without non-concurrent control, or under different models of the time train. Next slide, please. So when and how to use non-concurrent control, so it's clear that there is a benefit-risk balance. Again, a potential gain in power versus type 1 error inflation. And this is summarized in the table that you have here on this slide. So in phase 2, it's very much sponsor risk, and we can take in some ways the decision we want. In phase 3, when we are governed by the regulatory mandate, I think we need to separate what we would call a more standard experimental situation to a more difficult experimental situation. So we all have in mind, for example, rare diseases, but we could have in the discussion maybe other areas that can be considered as more challenging. And clearly, in a standard setting, I think regulators will ask probably to have a type 1 error control at 5%, whereas in a more difficult experiment situation, they are more prone, I think, now to move a little away from that, but I'm sure we'll hear from them on that topic. Next slide, please. And then, so finally, a few points that came out for discussion that maybe we'll have the opportunity to cover. So we talked about estimates, and really, should the estimate specify the use of non-concurrent controls, or is it a matter of estimation? Should the fact potentially that the control arm can change over time should be mentioned in the estimate? Another point that we mentioned has been discussed is the similarity with the use of historical controls. Although, of course, here we are more likely to follow the same procedures. Inclusion exclusion criteria can be the same centers, same investigators. And a good thing, of course, is that we have IPD in platform trial. We can help us maximize the comparability between concurrent and non-concurrent controls. And then finally, last topic, again, regulatory engagement, very important for us in the pharmaceutical industry. Talking to them early will be fundamental. Very important to pre-specify our assumptions, specifically around time trends. Which model do we use? How likely or unlikely these assumptions are? And then discussing, of course, the impact on type 1 error. There is probably also some room for having interim analysis in order to check our assumption and see maybe how we can react to that and at which costs. Again, importance on having different estimates and sensitivity analysis. And specifically, I'm calling out for the need of having a framework in which we could discuss that with regulators at scientific advice with the right information. Again, assumptions, operating characteristics, and sensitivity analysis. So I think that's all for the industry. Again, I don't think anything is really specific to our world, but very much global scientific discussion. So thank you.
Thank you, Olivier. Next, we will have a series of 3 short talks giving a health authority perspective. First will be Andrew Thompson from the EMA.
Hello, everybody. Thanks for the opportunity to present a regulatory review from the EU. Next slide, please. So I want to begin by saying thank you to the previous presenters for some excellent presentations with some good food for thought. I particularly liked Olivier's table there when he talked about the risk and who was taking the risk. And I do think the focus for me is on phase 3 studies, which are the pivotal evidence for licensure, and there won't be any additional data afterwards. And fundamentally, we do need to remember that non-concurrent controls are non-randomized comparisons, or at the very least, in terminology-wise, not fully randomized comparisons. And when people think of non-randomized comparisons, they often think in terms of RCT versus observational data, and that's clearly not the case here. Now, there's a sliding scale based on the information that's available. So, for example, if you had a platform trial that had 500 versus 500 subjects in your control and your active arm, and you borrowed 1 additional non-concurrent control patient, that's clearly a very different experimental situation to one where you might have 20 versus 20, but then you needed to borrow 50 extra non-concurrent controls. And the problem we have here is that both these situations, if the non-concurrent data is pivotal to the evidence base and to the decision that needs to be made from a regulatory perspective, there's a bit of a problem, because if you just need 1 extra patient to tip you over from being positive to negative in the first example, that's not ideal. And in the latter, you're probably not in a rare disease if you've got a platform where you can generate so many extra non-concurrent controls. And so it's likely that the experimental situation is such that a full RCT would have been expected. Next slide, please. And again, with non-randomized comparisons, sometimes they agree, and there's great work that's been done by people out there saying, these are the situations that we could look into in particular therapeutic areas when non-randomized data can be compared to randomized data using different data sources and seeing what are the situations in when they agree. And sometimes they just don't agree. And I don't expect you to be able to see the meta-analysis on the right-hand side, but it's for hydroxychloroquine, and all the trials at the top are the observational studies, and all the ones at the bottom are the randomized studies. And you can see that the non-randomized studies showed a positive effect when they're meta-analyzed, the RCT showed no effect at all whatsoever. And when you put together non-randomized and randomized evidence, actually you end up with some kind of effect you see overall, which probably isn't indicative of the true effect that's best measured with a randomized trial of no effect. Next slide, please. And yes, that's a fair criticism. I'm comparing observational data with trials, and actually we're not just talking about observational data, but non-concurrent controls, which is different. But trials themselves throw up surprises when we analyze them. A very interesting recent example has been molnupiravir, which is oral treatment for COVID-19. Next slide, please. We have data before and after an interim analysis for this. And the endpoint is risk of hospitalization or any cause of death through day 29. And at the interim analysis, where the trial was, well, the data from the trial was announced to the outside world, it was 7.3 versus 14.1, so very large treatment effect. And in the final analysis, that had changed to 6.8 versus 9.7. And both these analyses are statistically significant, okay? And a back of the envelope calculation just for the second part of the trial, so the data generated after the interim analysis, there's one treatment, there's one drug, nothing should have changed apart from time, there's no adaptations. You see 6.2 on molnupiravir and only 4.7 on the control arm. So clearly something is going on in these two different parts of the trial, and it seems to be, at first sight at least, that there are differences between the two different parts of the control arm before and after the interim analysis, even though nothing fundamentally changed in the trial. Next slide, please. So in conclusion, just because one is operating a trial under the same protocol, that does not guarantee that you get the same results out of it. Indeed, you can see very different results that are very different and perhaps difficult to interpret. If you see consistency across time periods in one arm, such as we saw in the molnupiravir arm, that does not guarantee any consistency of treatment effect across time periods, treatment differences. We did not see that here. And we need to properly understand features as to why this could happen. And ultimately, of course, infectious diseases in a pandemic may not always be the best use case. I think it's fair to say that non-concurrent controls recruited on the same protocol are likely the best option compared to, say, real-world evidence or control arms from other trials when it's acceptable to regulators. But please do not underestimate how hard it can be to convince regulators that it's acceptable to move away from a fully randomized design. Thank you very much.
Thank you, Andrew. Next, we'll have Daniel Rubin provide the FDA perspective.
All right, thank you. I'll give a few remarks on the FDA thinking on this topic. Could you go to the next slide, please? Ordinarily, our recommendation is that the primary analysis should restrict to a fully concurrently randomized comparison. However, if sponsors plan to leverage non-concurrent control data, then we think that a compelling rationale can and should be provided. Go to the next slide, please. The main advantage of a concurrently randomized control is that it provides the strongest guarantee against confounding. And such confounding could be caused, for instance, as we've heard, by temporal shifts in patient characteristics, trial conduct, background standards of care, and so forth, especially for a long-running platform trial in a rapidly changing setting, such as COVID-19. And in the presence of such temporal shifts, non-concurrent control data could potentially lead to bias or inflation of error probabilities, even if attempts are made to account for potential trends in the analysis. And one additional remark I should probably make is I know that a lot of the discussion today has been about temporal issues, but very similar considerations would arise in a platform trial if there was use of concurrent but non-randomized controls, such as if only some drugs were available at some study sites. Next slide, please. So what should a rationale for use of non-concurrent controls involve? We think that the rationale should discuss the likelihood of temporal changes during the trial, the feasibility of relying on only fully concurrently randomized comparisons, the extent to which non-concurrent controls will be used, the calendar time of separation between arms that will be compared in the trial, and the ability and underlying assumptions of statistical methods to account for potential temporal changes. Next slide, please. Now, there are some additional considerations and complexities that arise in a platform trial that wouldn't arise in a more traditional trial related to pre-specification. So for instance, if results were disseminated for a poorly performing control arm and another sponsor saw this and wanted to enter its drug into the trial to compare to those control data, that would be a potential data-driven random high issue that could potentially introduce bias. Could you go to the next slide, please? So what are some measures that can mitigate potential confounding? Well, as we've heard, one advantage that we have with master protocols that we don't have in historical studies is that we can try to ensure uniformity in the design and conduct, and especially the outcome. We think the master protocol should also specify a collection of baseline and post-baseline prognostic factors. This can help evaluate the comparability of treatment groups and allow for adjustment for confounding when and as appropriate. And then, as I've mentioned, and I think we'll get into more in the discussion, ideally the analysis should be pre-specified to the extent possible, with the recognition that there are some complexities with doing this in master protocols. And then could you go to my next slide, please? And finally, there are some additional considerations if using non-concurrent controls. We do think that the primary analysis should attempt to adjust for confounding. It probably shouldn't be just pretending that we're in a fully randomized comparison when we're not. And as we've heard, there are different methods for doing this. This can involve models that adjust for a function of calendar time, methods based on a network meta-analysis, and so forth. But no matter what model is used, we think that the underlying assumptions of the analysis should be described, and the operating characteristics should be evaluated under different hypothetical settings, such as temporal shifts. And finally, we do think that this is an appropriate setting for sensitivity analysis, particularly the sensitivity analysis that restricts to the unbiased but possibly noisy, fully randomized comparison. Thank you. I'll stop here.
Thank you, Daniel, for the excellent presentation. Next, Julia Superior will give the MHRA perspective.
Yes, I'm here. Don't panic, everybody. I had some technical problems. There we go. So I start, even though I can't switch my camera on, and I'm very, oh, there we go. I'm here. Hooray. Typical. Okay, so I have to start by saying if I could have the control, that would be great. But if not, can you move to the next slide, please, and say that the views expressed are my own and not those of the MHRA. Next slide, please. Yes. So we're all here today because we're not interested in comparing apples and pears, but what we're talking about is whether it's okay to compare the green apple to the red apple. So there's nothing earth-shattering here. I'm just reiterating really what Andrew and Daniel have already said, and showing you another picture of a platform trial. We're going to end up having 2 treatments in this platform trial, and obviously the controls carry on. You carry on recruiting control patients to the control arm throughout the duration of the trial. You have equal randomization, and these are the comparisons that we have. So the concurrent controls for treatment A are enclosed within the dark blue box, and the concurrent controls for treatment B are now enclosed in this new dark blue box, and the shaded out patients are not involved in those calculations. Now, some of you are ahead of me on this because I've seen some questions about it in the Q&A. What happens if there are different eligibility criteria for treatments A and B? So in this scenario, everybody that enters the trial can be randomized to treatment A. Everyone's eligible for treatment A, but but treatment B is contraindicated for some people. So that means that the nature of the controlled group changes in that situation. So because everybody can be randomized to treatment A or control, the comparison for treatment A is as we saw before. But when you are looking at the treatment effect for treatment B, because not all patients are eligible for treatment B, the control group is restricted in that case. So you have a smaller pool of control patients to look for. So this was referred to, Daniel referred to this in his presentation as well. So when you're talking about concurrent controls, sort of concurrency is necessary in inverted commas, but not sufficient. You have to look at other aspects of comparability, like eligibility for the treatments. And I would reiterate what Andrew said as well, is that okay, non-concurrent controls can be acceptable in the regulatory setting, but you have to convince us that it is acceptable, taking into account all of those things that we've already discussed before. So, so the bottom line is you have to convince us that the apples are similar enough that we're happy to compare them. And apologies again for the technical problems. Thank you very much. That's where I'll finish.
Thank you, Julia. Next, we will have a perspective from health technology assessors and payers by Ralph Bender.
Yeah, hello everybody. Here's Ralph from Cologne, Germany. Yeah, today is the day when usually the carnival starts in the streets of Cologne, but due to the known global time trend, this has not happened today, though it's good to have an alternative option like this interesting webinar. Next slide, please. Next slide, please. Due to the excellent presentations before, I have not very much to add. We all know why we need a control group. We want to know that if we see a difference between two groups, that this is caused by the test treatment and not caused by other factors. The old known guideline ICH E10 says that a concurrent control group is needed, and that test and control group should be similar to everything except of the treatment. Next slide, please. So we have heard that the use of non-concurrent controls, of course, can improve trial efficiency, but it's clear that if you do this, it violates the old ICH principles. It is prone to bias due to possible time trends. It requires untestable modeling assumption, and at the final end leads to increased uncertainty of the results. Next slide, please. So if you decide not to use non-concurrent controls and stay with the concurrent controls only, this has some advantages. You avoid difficult discussions with the regulators with unclear result. It allows the use of standard hypothesis testing, avoids subjective assessment regarding the adequacy of the applied modeling approach, the adequacy of type 1 error control, and the adequacy of bias reduction. At the final end, it allows clear-cut decisions without difficult discussions. So if you want to avoid difficult discussions, then avoid to use non-concurrent controls. But of course, next slide, please. There are situations where non-concurrent controls may be considered. situation with high medical need, when there is a need for a rapid, broad introduction of a new intervention, in the case of rare diseases with limited availability of control patients. But these arguments should not be overused. You can say nearly all the time that there is a high medical need, and this should be reserved for special situations. Next slide, please. So if you decide to use non-concurrent controls, there are some advantageous terms which can help to be successful at the final end. Of course, as the time period between concurrent and non-concurrent controls is short, it's a good feature. You need good arguments for the choice of the modeling approach. And yeah, at the very least, you should also additional concurrent controls, because then you have the possibility to assess homogeneity between the two kinds of controls. The effect can be estimated with and without non-concurrent controls. And in case you see that indeed the two kinds of controls are different, then you have the chance to use only the concurrent controls for the final analysis. And it's advantageous to have a large effect estimate to compensate for the increased uncertainty. Because if you can argue that even if there is some bias, this large effect estimate, it's not plausible that such a large effect estimate is observed by bias alone, then you have a good argument at hand to get a successful result at the final end. Next slide, please. So conclusion, if you want clear-cut decisions without difficult discussions, avoid the use of non-concurrent controls. If you decide to use non-concurrent controls, you should also plan concurrent controls, and it's advantageous to observe a large effect estimate. Thank you for your attention.
Thank you, Ralph. Next, an academic perspective will be given by Stevenson.
Can you hear me? Good. Okay, so first of all, I'd like to thank my academic colleagues for their input, and secondly, to absolve them of all responsibility of what I have to say in case I say anything stupid. Next slide, please. So just to give a historical overview, we've had these sort of issues before. Back in the 1920s and 1930s, Fisher and Yates started looking at incomplete block designs and discovered that in actual fact, treating the block effects as fixed was not fully efficient. There was interblock information to recover. But their randomization had taken between and within blocks. There's an extensive theory of this particular area, which perhaps should be better known to medical statisticians. Starting around the mid-70s, the meta-analysis became popular, and there what happened was that the trial effects were treated as fixed, but there hadn't been randomization between trials, only within. Use of historical controls started becoming more fashionable again in the mid-noughties, and there was a lot of important Bayesian work in which essentially the trial effect was treated as random and a certain degree of exchangeability was assumed. And then maybe more recently, we've had platform trials. We could perhaps push back the beginning of those a little bit earlier than that, in which there are differences between periods, and this can be overcome by modeling to some extent, as has been already said. Just one particular point perhaps worth mentioning here, it's not just a difference between time. If platform trials, for example, permit centers to join and leave, then there can also be a difference between centers. Next slide, please. So consequences of fitting a factor in a model. It depends when you fit a factor. The following effects occur. You can correct for bias. But if the factor is unbalanced, then inevitably there is some sort of penalty for non-orthogonality which occurs. So there will be some inflation of the variance, this in the sometimes referred to as the design effect. On the other hand, to the extent to which the factor is predictive, for example, if there is a difference between eras as regards time trends and you fit them, then in that case, the mean square error will be reduced. And so you get a benefit for standard errors. But there's also an adverse effect on second order precision. You start using up degrees of freedom. And if you're working in rare diseases, which was an area I was looking at fairly recently, then that can be a serious point. Next slide, please. So optimal approach. Well, in practice, a lot of applied statistics boils down to recognizing that there's a bias variance trade-off. Elimination of bias is paramount where one is information rich. It then dominates. So typically that's a situation in phase 3, or where credibility is essential. Again, typically situation in phase 3. And also where inter-study and inter-era differences may be expected to be large. So COVID vaccine trials are an example. In my opinion, it would have been disastrous to have used non-concurrent controls for such trials, but it's not the only case. It's been observed, for example, in trials in HIV, that patients who are recruited later have generally done better in terms of prognosis than patients who were recruited earlier. Where does one go down on the side of reducing variance? Well, in exploratory studies, rare diseases, phase 2 generally, and also where inter-study and inter-era differences may be expected to be small, chronic inflammatory disease might be an example. But also, I think something which people tend to overlook is blinding. If blinding is important, then this is only fully achieved by using concurrent controls. To the extent that you do not respect concurrent controls, then in that case, blinding is an issue. Next slide, please. So recommendations that we make, I think, essentially match many of the ones that we've had so far. One should be explicit about what assumptions are being made. One should be cautious about making claims for gains in efficiency. In my opinion, a lot of the early statements that were made about platform trials insufficiently pointed out the problems, and maybe regulators were playing catch up there because they were blinded by some of the hype that was put out. Be alert for different sources of bias. Difference of gene centers are important. Effects of time. It's not just a question of changing patients. It's changing disease. Think of new variants. Changing standards of care. Think of new personnel, even if the centers haven't changed, and so forth. And weakening of blinding. And above all, what's important is that all interested parties should understand the issues. Thank you.
Thank you, Stephen. I believe you will also give
Well, I will attempt. Unfortunately, Robert is undergoing heart surgery at the moment and can't be with us. So it'd be much better if he could have spoken to his slides. But I think we'll just put them up, and I won't try to say too much about them. So next slide, please. So I think what Robert wants to remind us for is that also at the center of this should be patients. And there are certain indications, certain problems where it is important to try and get treatments out to patients. And also another thing which people sometimes forget is that if you spend all your resources on large trials, then effectively there are a number of trials which have no patients on them whatsoever. Those are the trials you would have run. And in some cases, you may be forced into making decisions anyway for these particular patients. So it is sometimes misleading if one only concentrates on the evidence for what one managed, for the treatments one managed to study. Next slide, please. So this is the risk of bias, of course, for those patients that have drugs approved for their particular disease, and there's reduced confidence. And there's also the problem that if you enroll in such a trial, you may feel in the end that it's less likely to be definitive, and that, therefore maybe your sacrifice has not been as valuable as it would be under other circumstances. There are various sources of bias one should watch out for. Stage migration is one of them, which is perhaps underestimated. And just to say that the risk of bias can be reduced, but it can't be completely eliminated. Next slide, please. So recommendations, protecting against bias is most important in phase 3. I think all the speakers have more or less recognized that. However, there are other circumstances in which one might consider non-concurrent controls or other alternative control data. There are also different types of quantitative decision analysis which might be considered. And Robert finishes by recommending that some sort of qualitative decision tree could be used. I'm not competent to go into that. Next slide, please. I think that's the end.
Thank you to all the speakers. I think next, before we go into the panel discussion, we have a few poll questions we would like to ask the audience. Can we bring up the poll questions, please? Question 1 is, what type of organization are you affiliated with? I'm not sure. Are we going to show the questions at the end, or we can show them after each one?
It's just answers are coming in now, so.
Okay.
We have right now 35% participation. It's going up still, so
okay.
That's good. Okay, I think it's we approached the final the interim analysis. 60% have participated, but answers are coming in very slowly now. So I would end the poll for this question here and move to the next question.
Okay. The next question is. Oh, okay. Okay.
The others are all okay. We have all the questions have been answered. So the second one is,
does your organization have a platform trial planned, ongoing, or completed? Okay. We will see how the views on non-concurrent controls change after we have the panel discussion. So perhaps to give people a little bit of time to refresh, I propose that we take a 10-minute break, and we will come back again at 11:42 and start the panel. Welcome back to everyone. I hope everyone was able to stretch their legs and get recaffeinated so we can continue with a very exciting panel discussion. We have an excellent panel that covers industry, academia.
Yeah, and may I just add, there's one question in the Q&A to Stephen Sen. Stephen, if you could answer this.
Yeah, basically, I'm skeptical that by conditioning on patient-level covariates that one can control for all effects. Typically, I think that the center effects are overlooked. And if you think about cluster randomized trials, then no regulator would accept a cluster randomized trial as if it were a parallel group trial simply because you would include an analysis of covariance.
Great.
Stephen, I think Martin Mentz has a question to you in the Q&A tool. You find it on the bottom of the Zoom app.
Yeah, I thought that was the one from Stefan, but was there another one? Sorry. Stefan Ballestedt?
Ah, yes, it was. Sorry.
That's the one I thought I answered.
You did, Stephen. Thank you.
Yeah.
So let's go to the first question, please. So the first question we will discuss today are non-concurrent controls actually more reliable than historical controls? And this will be discussed by Lisa Hampson from Novartis and Matt Seidel.
Thanks, Peter. Maybe we can go to the next slide. So for reasons that several of the previous presenters have touched on, I think my answer to this question is, in general, yes, I do think non-concurrent controls from a platform trial are more reliable than historical controls. And I think, as several presenters have argued, generating non-concurrent and concurrent controls under the same protocol should increase the likelihood of comparability and reduce the risk of bias. After all, our key efficacy endpoints are consistently defined and assessed. We collect data and can handle in a consistent way key intercurrent events. Our controls are drawn from the same centers, from the same investigators, and our patients are sort of fulfilling the same inclusion-exclusion criteria. Clearly, within those bounds, there still could be some drift over time in the patient population. But usually when we have non-concurrent controls in the platform, we will have access to their individual patient data, in contrast to historical controls where we may only have access to aggregate level data. And if we do have that individual patient data, including information on baseline prognostic factors, that may help us to characterize changes in the patient population over time and potentially address those in any analysis. But I'm conscious of the comment from Andrew that the same protocol does not guarantee the same results. And I completely agree. There is still a risk with non-concurrent controls that there could be time trends, including the fact that our standard of care could be changing over time in a way that's difficult to characterize or quantify, and our patient population could be changing with regards to baseline covariates that we simply haven't measured. And so we can't account for those, as well as the fact that the disease could be changing. So with an infectious disease, the variant of the virus could be changing. Next slide. So I guess we might ask ourselves, are there any proactive steps we can take to really minimize the potential impacts of time trends? And I think one approach could be to try and plan ahead as far as possible to capture elements or data that may help us characterize or model out in the future a potential time trend. So all potential key prognostic factors. And as far as is feasible, recruit from the same centers and regions throughout the trial. And we may try and predict ahead of time sort of the likely size of potential time trends. And clearly this would need to, whether this is even feasible, would need to be evaluated on a case-by-case basis. But I think discussions with our interdisciplinary clinical trial teams, particularly with our clinical colleagues, would help us to understand the potential risk of time trends. And if we do have some external historical controls available to us, some exploratory analyses to see if there are time trends in those may help. The absence of time trends might not guarantee there won't be any in our platform trial, but at least it might provide some reassurance that the disease and the treatment landscape for standard of care is relatively stable over time. So I feel optimistic that non-concurrent controls in the platform are more reliable than historical external controls.
So I feel sympathetic to everything that Lisa said there, but I feel a bit more cautious about whether non-concurrent controls are going to be very useful in a lot of instances. Let me give you an example of some data that came out from the Stampede trial, a large platform protocol in prostate cancer. These are the data that came out of one of the first results. It's phase 3. It's looking at survival, just comparing concurrent patients allocated to the control arm, the top yellow grouping, or the patients allocated to add chemotherapy to the standard of care. So adding docetaxel, what we showed in those patients allocated to this comparison, that's just short of 1,800 patients, was a hazard ratio of 0.78. This was both statistically significant and clinically meaningful, an improvement to adding chemotherapy. Could we go on to the next slide? The first added comparison in the Stampede platform looked at adding abiraterone to the same standard of care that docetaxel was being assessed against. Those patients were randomized later, between late 2011 and early 2014. And there, when we presented the results, we saw a hazard ratio of 0.63, again, statistically significant and clinically meaningful. And the clinical community was jumping up and down with excitement on this one because it was a much bigger effect over control than docetaxel had shown. If we go on to the next slide, we thought we could look at a head-to-head comparison for those patients who have been contemporaneously or concurrently allocated to either of those two research treatments. That had 566 patients in it. So that's not a small number. It's bigger than the first couple of trials I worked on when I started out in trials many years ago. This is just those patients allocated to either of those research treatments between late 2011, early 2013. If we go on, we'll see the population is perhaps a little bit more similar to the docetaxel comparison than the abiraterone comparison. I'll mention why in a second. And when we look at the treatment effect, what we saw was no evidence of a difference. If anything, the treatment effect pulled a little towards docetaxel rather than abiraterone, but it's nothing conclusive here. So let's go on to the next slide. Our sense here was that, we thought that comparing concurrent patients was going to be the cleanest head-to-head assessment of these treatments, and I still maintain that it is. But that finding was a surprise to the clinical community, having seen the two direct results against control. So why is this? Well, partly the population recruited to any trial may shift over time, even with the same eligibility criteria. And that happened a little bit here. When we stopped recruiting to the docetaxel comparison, when we stopped having a chemotherapy arm, people became a little bit more willing to put in their older patients, even though the eligibility criteria hadn't changed. And we also had another arm that had been added looking at only metastatic patients. And so while later on overlapped more with the abiraterone comparison. So while the abiraterone arm and the control patients were fair across, they did have proportionately metastatic patients going in over time. Also worth noting is that something that perhaps is much more difficult to measure, the treatments available to people at relapse changed considerably with results of other trials as time went on. So we think, that looking at indirect comparisons, even from within the same protocol, can be misleading. So it just makes me feel really rather cautious in using non-concurrent controls within the same protocol. There's a longer version of this if you want a bit more detail that we put online. There's a link there and we'll put that out later on.
Thank you, Matt. Is there anyone else from the panelists who would like to comment on this question?
Sorry, so this is Kurt Feely. So, to start off, I agree with the comments made by Lisa, others beforehand. I certainly think non-concurrent controls are vastly superior to historical data and real-world evidence. I think one of the important parts here is one lesson from historical data is along the lines of what Stephen Sen was saying, the ability to quantify the bias variance trade-off and actually say, we know there's an assumption, there's a level of time varying effect that we can tolerate and still win the bias variance trade-off. And we should certainly quantify what that is. We need to win it on average, so to speak, to use it. And certainly some indications are going to be more favorable for non-concurrent controls than not. I also think one of the key things is a difference here is that there's a lot more ability to do sensitivities in historical data. You've got a lot more information than one arm in your current trial, and you're hoping it's close to history. And kind of to go back to that molnupiravir example, so that was a situation where the two time periods are very different. But in a platform, we wouldn't just have control and treatment. We would have all of the other arms. And I think it would be incredibly informative to be able to see, did those other arms, were they stable like molnupiravir, or were they vastly different like the placebo appeared to be? I think those kind of sensitivities, they're available and they can be done. And, the examples that were shown, we wouldn't make a mistake of using, if that were a non-concurrent controls example, we would catch it in the sensitivities and we'd do the right thing. The final thing I'll say on this is, I found it interesting. The molnupiravir example is very, very complex and we don't know what to do. But this is a situation where there was this, perfect one-to-one randomization, and it also didn't solve the problem. So I think there's always a sense in innovation. We should always know what the assumptions are. We should know what the risks are, but we should also recognize that not the other solutions are not perfect in this setting either. Thank you.
Yes, thanks. So let's move on to the next question, which is on the methods to use to adjust for time trends. So are there scenarios where we could simply pool concurrent and non-concurrent controls if we should use these model-based approaches, and how smooth can we assume the time trend to be? Are there maybe other methods that we have not talked about? And we'll have James Wason and Kat Feely reply to these questions. So
yes,
James
thanks
Wason
very much.
Newcastle University.
Thank you, Martin. Yes, so my perspective is from someone who's mostly interested in chronic inflammatory conditions, which are often pretty rare, and it's hard to do large, fully powered randomized trials. And often they're quite stable over time, although in terms of outcomes, having said that though, some of them have been quite affected by COVID, so maybe that's not been true over the last couple of years. But I'm confident what I say might sound a bit different if I work more in common infectious diseases where things are quite different. So in the types of settings I'm interested in, the reason for time trends would probably be more likely to be different patients being enrolled in the trial over the course. And this is something that we can observe in the trial and potentially correct for, as opposed to, say, the standard of care changing over time. Having said that, though, when you start a trial and when you open new centres, you may have a kind of list of patients who have had the condition for a while, where you have a lot of established disease patients who are recruited earlier in the trial or earlier when a centre's open. and then more newly diagnosed patients subsequently later in the trial. So that could be something relevant to more chronic conditions. So I really think that it's been very interesting to hear all of the talks, and thanks everyone for the great talks. And compared to when I was thinking more about this with work with Kim Lee a few years ago, I think there has been some progress made, and it's nice to see all of the new work coming out with new approaches. I still think fundamentally, when we are we're using modeling approaches, we do have to make the bias variance trade-off that we've heard about. So the more restrictions we place on how we're modeling how things are changing over the course of the trial, the more power we can gain, but the more chance we have for bias and type 1 error rate control if we get it wrong. And that very much agrees with what we found back in our BMC medical research methodology paper. I think the Bayesian approaches are really quite appealing. Nice to hear about the Bayesian time machine, which is a good name. I think also we could use Bayesian approaches to potentially include not just data from in the trial, but if we had independent data on how outcomes are changing in, say, registry data and such like, a Bayesian approach seems like a great way of incorporating that and being more flexible but informative with modeling trends. So I think that's a promising thing to explore more, and a lot of areas for future research. For rarer diseases, which I'm particularly interested in, I do think if you talk to patients that they're willing to accept kind of risk of bias in trials in exchange for increased power and making testing more treatments feasible. And maybe we should be thinking more about patients when we're thinking about the types of methods that we're using to make decisions to treat them. So I think I'll stop there.
So this is Kurt. I certainly agree with all that. Always good to go second. I think that in terms of the methodology, I think this is a pretty open field. I think the unsmoothed models are often easier to explain to people, and it's easier to demonstrate where the bias assumptions are. The smoothers have the advantage that essentially your sample size savings, your increase in efficiency goes up a notch. So you get another 10 to 15% savings out of that. And I'd love to see more work in this area, not just on the time machine, but I know there's work from Lorenzo Trippo's group on using generalized additive models. I think all of this is going to be an active area going forward on what exactly is the optimizes that bias variance trade-off.
Thanks a lot. Yevgen?
Yeah, hi, I hope you can hear me. Just to put, first of all, many thanks for the fantastic, outstanding presentation. I think at least in my head, it's been clear how maybe approach the challenges and analysis method. And Martha's presentation is actually put really, really clear case, at least to me, that the real challenge is interaction. So the time drift itself, we can handle. And Scott's presentation is fantastic, showing, okay, we can handle essentially any drift, given that it's kind of proportionally affect each treatment arm. The challenge is this treatment by period interaction, or however we call it. That's where, unfortunately, not much could be done analytically. But then I hope still, like, collecting data, analyzing, as a Stampede example that Matt put together, it's fantastic in a sense that, okay, we collected the data, we saw something, and then because we have data collected in a consistent way, we managed to explain what happens, or at least to put some conjecture. I think Andrew also brought the point that it would be nice to understand real mechanism. And maybe bridging a little bit to the first question, data collection and platform trial absolutely gives this opportunity. Historical data, not much. Essentially, we just facing maybe some difference and then theoretical speculation why it might happen. Just a little bit to contribute to this discussion.
Yeah, thanks. Ursula?
Yeah, I think what I like as analysis methods is methods that allow interpretation what the time trend actually is. So just a blind adjustment can always overcompensate a bias and create a new bias. So to me, it would be essential that within the method or with accompanying methodology, you try to explain the presence of the time trend by something, because that to me would increase the reliability of the results.
Thanks. And Olivier.
Yes, thank you. I just wanted to bring a sort of sub-question on that on that topic that Peter brought when we prepared the industry perspective, because that very much relates to the methods. Is the importance of interim analysis to cross-check the models that we can have pre-specified at the beginning. And I thought a little bit more about it since we talked about that, and that reminded me a bit. You know, the max combo and non-proportional hazards, where you have an interim and then you try to find the right methodology to test that. And I was just wanted to ask to the panelists if somebody has used that, you know, has looked at the interim, checked the time trend, changed the primary analysis, and if anybody has thought about the impact on type 1 error, because I think that would be where the issue would be into doing that. So just to trigger maybe that point.
Do we have any quick response to that from the panel? Andrew?
Yeah, thanks. I think it will. The underlying assumption is that there's a protocol element that is driving that, and that if you change the protocol, that you will see different results, and it might actually be for all the other reasons that we've discussed. But I think that centers learn how to implement protocols better the longer the trial goes on. So they do change over time as well. Stephen made an excellent point about site personnel changing being potentially something, because if you bring in new people to an established center, but they don't know the protocol as well as the people who've left, then that can impact the results. So I would just be wary of automatically assuming that there is some magic amendment that you can make to your protocol to make these time trends either disappear or be affected, whereas it may just be, for example, a factor of people implementing the protocol more efficiently as the trial progresses. Thanks.
Thanks. Okay, then let's move to the next question.
Are statistical adjustments sufficiently robust for pivotal analyses? This is the whole multiplicity topic is a hot topic in anytime we discuss platform trials, and there are many differing views. So this topic will be discussed by Yevgen and Stevenson.
Yeah, if you don't mind, I probably started with the first iteration. And I would like to pivot the question a bit, put in a clause sufficiently robust for the purpose of a situation. I hear there's some common consensus that NCC is quite possible in rare disease setting, and medical needs, pediatrics, etc. And B, it's rather extremely hard would be to come up with some convincing rationale for the use of NCC in conventional, two positive well-controlled studies for registration. But of course, we all know that there is a continuum of spectrum and FDA guidance document on what it takes to demonstrate substantial evidence depending on circumstances. I think it's a really good reference. Maybe to toss some couple examples, potential examples, when we can choose and inform ourselves and community about what design and what analysis options would be worth considering. And first row in this table, I just said, okay, it's quite common. It's often the case in oncology study where there is a medical need situation. The single arm study with objective response rate is viewed as an adequate evidence for accelerated or conditional approval. And then as more treatment becomes available, field mature, disease handled better, the level of evidence naturally can be raised, requiring a randomized head-to-head study. And I think it's reasonable to assume that somewhere in between this increment, NCC type of design would be appropriate. We're raising the bar, and we kind of recognize the strengths of the evidence, and that could be a niche for NCC use. Another example, also maybe more relevant to oncology, it's about establishing evidence regarding the overall survival drug effect. And again, very often PFS is viewed as acceptable primary endpoint. Of course, overall survival super important and will be studied as well, but it could be less powered. And one of the main reasons to have potential underpowered analysis is the crossover to the treatment that already established efficacy in terms of PFS improvement. And again, platform design potentially contribute a lot for the inference of regarding the OS, because pulling across several cohorts, or how we call them ISI, is really attractive from that perspective. Plus, very often OS considered as a key secondary, hence maybe this control of type 1 error a little bit already addressed from the safeguarding regarding, like, for example, PFS based on randomized data only. I have several points, but I really want to mention regarding the kind of several ideas about missing equipoise and the right approach for the general situation. I would elaborate on Professor Beckman's point on his slide, avoiding trial not funded. Again, using oncology setting example, several mechanisms prove the action, and then in the relapse refractory lines, really several options appear, and the combination of different mechanisms kind of piles up and multiplies very, very rapidly. Before you know, you have like really dozens of options to consider. But in the current paradigm, it would be almost impossible for the sponsor to follow all interesting potential collaborations. So we have to make priorities, and that means, unfortunately, some quite attractive combination of drug will not actually be started at all. And maybe again, that's where a platform could help us to kind of mitigate those lost opportunity in terms of getting evidence and study the field. I'll probably stop here for now. I want to leave more space for other panelists to discuss.
So I would just like to make the comment that I think that the distinction between Bayesian and frequentist methods is largely a red herring. It's not the inferential framework that matters, it's how you actually model the particular effects that you might be modeling. The strict frequentist view, of course, is that every time you change the allocation ratio, you should declare a new stratum. So the cost of doing that is that the imbalance that there is between groups is paid for. There's a non-orthogonality penalty. But from one point of view, if there could be a difference between eras or between whatever you're looking at, that's the right thing to be doing. You're paying a penalty you should be paying. I disagree with what Scott said, for example, about what normally happens in Bayesian meta-analysis. My experience is that the trial effect is not treated hierarchically. Usually what happens is an uninformative prior is used for each new trial that is added. So that would be the equivalent to what happens in a frequentist meta-analysis there. My own thinking was particularly influenced by the target study I took part in. I was a member of the Data Safety Monitoring Board. That was running 2 sub-studies. The protocol is as similar as you like for the 2 sub-studies, apart from the fact that the control treatment was different. This was in rheumatism, and the control treatment was naproxen in one case, and it was ibuprofen in another. The intervention was lumiracoxib. You can show clear differences between the groups at baseline if they're in different sub-studies. If they're in the same sub-study, there's no difference. But you can go further than that. You can compare lumiracoxib to lumiracoxib across sub-studies, and you will find significant differences at outcome between the 2 sub-studies, despite the fact that they're run at the same time under the same protocol. And the explanation probably is that different centers are involved. That's probably the explanation for this. So this again is a thing that people tend to overlook. And the point about analyses that respect randomization is they're very rarely optimal, but they're very rarely bad. And most alternative analyses other people propose cheat in some way because they smuggle in an assumption which is not on the table. I think we're gradually coming to see that these assumptions have to be on the table, and I welcome that.
Thank you, Stephen. Are there any other panelists who'd like to comment on this topic? I don't see any hands raised, so perhaps we can move on to the next question then.
Yes, and the next question is on the factors that determine the importance and acceptability of using non-concurrent controls. So what are the scenarios where we think that the benefits outweigh the risks of using non-concurrent controls, especially say in phase 3, what would be the requirements to make non-concurrent controls acceptable? And we have 3 panelists that will comment on that. It's Olga Machenko from Bayer, Florian Klingemüller from the Austrian National Competent Authority, AGES, and Matt Seist from University College London. So Olga, please go ahead.
Thank you, Martin. I will go first. As was already mentioned by many of our speakers today and panelists, inclusion of non-concurrent control and analysis is a question of variance bias trade-off, and examples of cases where reduction in variance is major concern were given by Stephen. For example, exploratory trials, rare diseases. There are also a number of cases where it's acknowledged already by regulators that strict type 1 error control at 5% is not possible. And we know such rare disease indications and also in some pediatric trials. In order for non-concurrent control to be considered for inclusion in analysis, non-concurrent control have to be randomized under the same protocol with the same inclusion-exclusion criteria, use the same assessment visits, procedures, data quality, possibly to use the same centers. We should plan for known sources of bias, effect of time, weakening of blinding by reporting interim results or results from graduated treatments, and pre-plan the corresponding mitigation strategy such that a time trend can be adequately modeled. The choice of endpoints matter, so use more objective endpoints probably would be more acceptable. Such trials, of course, exist. Platform trials like I-SPY 2, GBM Agile, Precision Promise have used concurrent and non-concurrent control to analyze data and make comparisons. And Scott already presented I-SPY 2 trial. It's one of the first platform trials, and already several treatments graduated and were even approved. What is interesting to me personally, that every time we discuss right now, we know regulators stand. They accept analysis with a common control, but they say we need to have a good convincing reasons to use non-concurrent control. What about the totality of evidence? What bothers me really that when we give the drug to patients after the approval, doctors are not checking eligibility criteria which was used in phase 3 trial. If we use only concurrent control and ignore absolutely everything what is happening in a trial, we're limiting ourselves. Why can't we actually, as a first pre-planned step, to look at the totality of the evidence and data, and then eliminate non-concurrent control if it's not appropriate to use and condition for the sound inference does not hold? And thank you very much.
Yeah, thank you. First, let me point out that these are my personal opinions and not those of my agency or any working party that I'm associated with. So I will talk more a bit about maybe possible requirements or reasons why we would not like to see non-concurrent controls. And I think there is not much that others have not said yet. So the first is that whether non-concurrent controls could be acceptable is certainly a consideration that needs to take the indication into consideration, and whether assumptions that are required are plausible cannot be decided in general from a methodological perspective. Another thing, and that's when we have seen two very interesting procedures how you can adjust for time trends and possible biases, is that in a benefit risk assessment, we do not only look at the primary endpoint for efficacy, but also other important endpoints, and especially safety, have a big impact. And so there would need to be also an assurance that comparisons between non-concurrent controls and the treatment do not affect these other parts of the evidence package as well. Now, we have seen a couple of examples how the pandemic has shown us that at least for the development of COVID-19 treatments, the use of non-concurrent controls should be generally discouraged, because the disease changes over time, the attack rate of the disease changes over time, and also the standard of care changes over time. But and I think James mentioned it as well. This is not only limited to treatments for COVID-19, but we have also seen that the pandemic has affected other trials because patients were not able to come to their study visits, or hospitals may have been overloaded, and this was changing over time. It was changing between regions. And I think it is a good example also how externalities that are not expected and can not be really planned for may have an impact on a trial, and it would create challenges if we have then comparisons to non-concurrent controls. Now, this is maybe a lazy argument, but assessing non-standard methods and discussing whether certain assumptions are plausible and can be assumed in a certain situation requires additional resources and also specialized expertise, not only on the sides of persons planning the trials, but also on the side of regulators that evaluate the results from these trials. And I think we would be willing to make this as extra effort if there's a clear benefit to the patients, but simply arguments that something is more efficient or speeds up development may not be enough. Now, a point on earlier stages is, of course, in earlier stages, all of this is maybe less controversial. But we should also not forget that also earlier stages in drug development act as gatekeepers that can have positive and negative outcomes if certain treatments may not be carried forward because there are no resources left, but also especially in situations when earlier stages are promoted to be pivotal trials because we perceive someone perceives that there is an overwhelming results. The biases that can be introduced can also be a problem there. Thank you.
Thanks, Florian. And the third speaker is, sorry for the mix-up earlier, it will be Max Palmer, who will present also from University College London.
Thank you very much. So I won't repeat much of what I've said, but reflecting on it, at least from the panelist perspective, I think there is an element of furious agreement that non-concurrent controls are probably not ready for prime time in phase 3 trials in most instances, whether they be platform or not. and non-concurrent controls might be useful and appropriately used in phase 2 trials. And part of that rationale for use there is because the variance is so large in phase 2 trials. We know their size is generally modest, and the variance bias trade-off may very well be worth it for the sponsor. I'd like to talk a little bit about one issue I don't think that's not been discussed in the phase 3 trial context, is the issue of sort of what I'm calling a logical inconsistency between doing a platform trial and using non-concurrent controls. And this is based on experience over now nearly 20 years of doing platform trials. Our first sort of major platform trial started in 2005, which was the Stampede trial, which Matt Sides has described to you in brief, and started in 2005. It's a phase 3 trial, it's still ongoing in 2021, so that's 16 years, and will go on for another 5 years at least. So that's probably 20 years that trial will be going, testing upwards of about a dozen different therapies, more than a dozen different therapies over that 20-year period in the phase 3 context. On the left-hand side of this slide, you'll see all of the trials that we've been heavily involved in, all the areas that we've been heavily involved in, which we've launched multi-arm, multi-stage platform protocols, because not all platform protocols are either multi-arm or have many stages. So all of these have multi-arms and multi-stages, and all are in the phase 3 context. And we've thought hard about the value of looking back at them, the value of looking at either non-concurrent controls, because a number of them are continuing over a long period of time, for example, Stampede, compared to using just concurrent controls. And one of the arguments that we've considered is that actually we could reduce the number of patients and the cost of some of these comparisons if we use non-concurrent controls. But the challenge we have here is that platform trials, I'm on to my second bullet point on the right here, platform trials quite clearly spread patients over a number of research arms. And we've already heard the argument that we need to use the non-concurrent controls to reduce the variance of each pairwise comparison. The problem that we've hit is that if a platform protocol has 5 concurrent research arms, let's say, quite reasonably, our reviewers, both on funding committees and when we approach the regulators, ask, why not, if your proposal is to use non-concurrent controls, why not you test fewer research arms, use concurrent controls, get an unbiased result, and reduce the variance for each comparison in that way? And that's a hard one to answer when you look hard at what are, on the left-hand side, both common and uncommon disease. Motor neuron disease, for example, lower down the list, isn't a very common disease. And consistently, we answer that it's better to test fewer research arms, get the unbiased result, if we want that result to be accepted by the community at large, rather than just ourselves, which includes not only reviewers and regulators, but of course, practitioners, patients, and the like further on. And the last thing you want when you publish a positive phase 3 trial is to then have enormous arguments in the literature of whether that is a real result or not, and whether it's applicable and robust and resilient. You don't want to spend all of that time and money doing that, and then find actually you're spending most of your subsequent time and money arguing the robustness and reliability of the result. So I think there is a real logical inconsistency between doing a platform trial, having a number of concurrent ongoing arms, and then setting a major argument to use non-concurrent controls. I don't think that argument has been so perhaps clearly made before. And whenever we've looked at it hard, we always come down on the fact that if we want to persuade the world that we've got a robust and resilient and reliable result, we use concurrent controls. Thank you very much.
I think Martin's muted.
Sorry, I would ask the panelists to give their responses in the chat, so you can respond to the comment from Max. And hand over to Peter.
Okay. Let's move on to the next question. So this is a fairly broad question. We want to think about some of the design operational issues that we may encounter in these type of designs with non-concurrent controls, and this will be handled. This will be discussed by Yevgen and Daniel.
This is Daniel. I can start, and I'd like to discuss two design and operational issues. The first has to do with the timing of pre-specification in a situation where you have an ongoing platform trial, potential use of non-concurrent controls, and a sponsor may not realize that it wants to enter its agent into the trial until it's ongoing, and there may be different sample sizes and safety assessments from agent to agent. Ideally, I think that first of all, the decision whether to use non-concurrent controls or not and the principal features of the analysis would be envisioned even before the start of the platform trial. And ideally, the details of estimation at the level of the SAP would be pre-specified before any assignments are made to that agent of interest. But that being said, I think it is hard to give across-the-board advice on timing of pre-specification because of the different eventualities that can occur in a platform trial. So I think it's useful to take a step back and ask, why is it the pre-specification is emphasized? Well, what we want to avoid are the types of forking paths or random highs that can come when there's a data-driven decision on what analysis to use. So I think regulators would be very skeptical if there was any type of selection of the non-concurrent control data where, say, there was a proposal to use data from time periods A, B, and C at sites X, Y, and Z, if those were chosen favorably from a larger pool of data. So the second design and operational issue that I wanted to discuss was related to sample size planning and sample size re-estimation in a situation where the weight of the non-concurrent control was not necessarily fixed, but might be based on the trial results itself and the similarity to concurrent controls. The issue here, I guess, is that if the non-concurrent controls are down-weighted, then the sample size that's anticipated at the design stage might be leading to less power than had been planned. But if there's a sample size re-estimation, then the alpha spending definitely has increased complexity if you're kind of involving this dynamic weight of non-concurrent control data. So in this situation, well, I guess first the comment I should probably make is that the Bayesian approach handles this fairly automatically from a purely Bayesian perspective. If the posterior probability of efficacy or futility is not high enough, then you would increase enrollment. In terms of frequentist operating characteristics, again, I'd like to take a step back and kind of channel Professor Sen's comments and others that this is a bias variance trade-off problem, and the use of non-concurrent controls in the first place might be appropriate in data scarce settings where we're concerned about variance and might be willing to make some statistical assumptions and give up kind of analytical control of bias and type 1 error control. So given that we're in that space, it might be appropriate to evaluate any type of sample size re-estimation approach through, I guess, a simulation based characterization of operating characteristics. So I'll stop there.
Yeah, I would echo maybe quite a few comments that Daniel already made, and just maybe dwell on a bit on this question. In terms of design and operation, what already been alluded is probably design uncertainty, and in particular on regard on the reliance of NCC. I think it would remain in the pragmatic trials and to what extent we can use NCC at the final analysis. Pre-planning is very important. I personally would prefer the design that, of course, we don't have uncertainty as to whether we can use or not to use, and maybe relying on some set of criteria. For example, POCAC in 1976 listed very, very nicely what we might want to check. in order to ensure that we have exchangeability. I don't know, another maybe points to consider reasonable strategy is to plan a platform study, recognizing that NCC might not be fully endorsed, and then at least make a minimum adequate power for each ISI, for each intervention cohort. So you have reasonable powered study, and then if NCC happens to be consistent and will pass the checks, then we can maybe improve power by pooling. It's a very interesting topic that I think in the area, and Daniel also mentioned, is try to use interim analysis in order to make this assertion, and then maybe alter in a pre-specified way how we collect the data. If we see that controls and other factors behaves reasonable and there is no surprises, then probably we can curtail a bit study duration and otherwise increase it. I think it's pretty much ongoing area of the research, and I expect much could be done and reviewed in that area. Very briefly to touch upon principle about pre-specification, I think it's very intuitive, easy to endorse by everyone. Of course, this issue of selecting random high or random low for the decision of using or not using, or even to enter platform, is a recognized issue. Again, it still would be nice to have a documented effect on that. It kind of, we all intuitively recognize it, but I'm not personally aware it's been quantified. And finally, last but not least, I really want to dwell a bit more on this individual patient level availability. I think it offers quite a bit of possibilities. Also, that forum been fantastic set of presentation, as I already mentioned, covering quite a few subjects, but maybe for the future meeting that we might want to have is like individual level patient data control, propensity score-based analysis, etc. might benefit, because the moment when we start using more and more of those platform trials, we'll learn, and the best way to learn is from data, how we can use baseline, multiple endpoint, post-randomization, disposition, safety, and essentially maybe even running like machine learning exercise. If you have two sets of controls from different periods and you really torture the data through machine learning or other techniques and they are exchangeable, that might add value. I'm trying to say, let's be optimistic. I think lots of issues that we are discussing today have been very vocal 10 years ago when we discussed application of adaptive design at the era when it was less well understood, and 10 years passed, and lots of them been resolved by just practice and getting more experience. Overall, thank you again for the excellent feedback and presentation.
Does anyone else from the panel like to comment?
Just make one comment. I think that what we should not lose sight of is the fact that platform trials can actually be very useful whether or not one recovers information from concurrent controls only. I mean, they can actually make the business of starting new therapies using a master protocol more efficient. I don't know whether any panel members know how the recovery trial has worked. This has been a very successful trial in therapies for COVID. As far as I'm aware, the principle of concurrent control has been maintained by that trial, but nevertheless, it has made it possible to study many, many treatments in a rapid and efficient way, as far as I'm aware.
Okay. We have one additional topic that has come up within the chat that we perhaps like the views of the panelists. It's related to sensitivity analysis, and what is the role of sensitivity analysis in the platform trial?
I'm sorry for interrupting. There seems to be arm raised by Max, and on the previous discussion, we might cut.
Yeah, so please,
I'm not sure if it's visible for you. I just want to make sure that we won't jump on one topic.
Please go. I missed it. Please go ahead, Max.
Oh yeah, sorry. So just to comment on Stephen Sen's point and the same point made by Olivia Collignon, the big benefits that we see of running platform studies in all those range of diseases in the phase 3 context is the common control arm and the operational efficiencies that you get. They're the two big benefits. that you get of running these agents. And one example is the Stampede trial, in which I indicated that we compared about a dozen new therapies over time, over the 20-year period it will run. And if we'd done that back-to-back as sequential trials, it would have taken us many, many decades, 50, 60, 70 decades, depending on how long each one took to fund, etc. So you can see the operational efficiencies are huge, and the statistical efficiency is gained through the use of the common control arm.
So the additional question that's come up during the course of the meeting that we perhaps like the panelists to comment on is can we ask what is the role of sensitivity analysis in platform trials? As long as we pre-specify, could one present both concurrent, non-concurrent, and as long as we specify which is main, which is sensitivity, and why? Would anyone like to tackle this?
That's it.
James? Go ahead.
Sorry.
I think I was just going to basically make the same points I put in the chat about, I think sensitivity analysis is really important. So we're in the phase 2 setting, and we, in principle, agree that NCC could be useful. Then what do we do if we get quite different results? So that could be same point estimate, but narrower confidence interval with the NCC, or it could be quite different point estimates. Which do we believe?
Max, would you like to comment further?
Yeah, I think it is useful to distinguish between the phase 2 and the phase 3. I think for the phase 3, for the phase 2 component, it probably matters less what your primary analysis and what your sensitivity analyses are. And if you get small differences in the sensitivity analyses, then, because it's largely the sponsor's risk which one they choose to believe, it matters less which you regard as primary and how many analyses you do, I think, because what you're trying to see is for what risk you're taking about going into a phase 3 trial. In the phase 3 context, it's much more challenging, I think. Even if you get small differences or modest differences in sensitivity analysis, or decide on the basis of those whether you use non-concurrent or concurrent controls only, how close are results, how close do they have to be before they're considered to be similar? I mean, that's just a finger in the air question, isn't it? And so that question just cannot be answered in any sensible way. So in the phase 3 context, yes, you can do sensitivity analysis, and if they all line up with your primary analysis in terms of being statistically significant, you're fine. But if you're using that as a basis to combine data, particularly concurrent and non-concurrent controls, I think that's a really dangerous thing to do, because you don't know what is similar enough to justify combination.
Okay. Scott, then Andrew, then Olivier.
Thanks. So there's no question that sensitivity analyses around the different patients are really important, and they're different sensitivity analyses in a platform trial because of the overlapping arms. I just the sense though that this isn't ready for phase 3, I just completely disagree. It doesn't mean in every circumstance it should be used. And the 2.5 hours have been caution after caution after caution, which statisticians are really good at. But it's ready for phase 3. It's better answers. When the sensitivity analyses give you different answers, it may very well be the use of the non-concurrent controls is exactly giving you a better answer. So it's not as though one is better at all, but it's where we need further scientific investigation into them, which is the importance of those sensitivity analyses.
Great. Andrew.
Thanks. Yeah, so a lot of regulatory decision making is making binary decisions. Do we license this drug, yes or no? And ultimately, in order to license something, we have to have some kind of way of discriminating between products that we license and products that we don't. And so we like to pre-specify things and hold people to them. And I find that the problem with this would be in the situation where the sensitivity analysis of either way offers you something different to what your main analysis is offering you. And that's always the regulatory problem. It's always at the margins and never when it's clearly succeeded or clearly failed, and then we can all be happy, tick the box and move on. And it's those margins where whatever the methodological question we're asking is where the problems arise. And so the question is, which of these should be prime, or which of these would I be likely to more favor? Well, I'm sorry, it would be the concurrently controlled analysis, because that's one I would want to put more weight on, because I believe it's an unbiased estimate of something that I'm interested in. And I would have concerns that the one that included the non-concurrent controls could be a biased estimate. However, I'd like to add a caveat to that, and I think it's important to reflect that there is a fundamental difference between a platform trial in a very common disease with multiple hundreds of patients who could be enrolled, and the differences observed between these different control arm rates versus a platform in a very rare disease where companies have come together to work together to set up a platform, and they've got a large majority of patients that are actually available to them full stop in the world. And in this situation, if we see these concurrent controls are actually similar to the non-concurrent controls, but you've just missed statistical significance, and then adding them in to augment the control arm tips it over the balance to the p equals 0.05 level. I don't really see a fundamental difference there, and I would be happy to weigh up both the concurrent and the non-concurrent control analyses as a part of a holistic view. But I think we have to be realistic that the experimental setting with the availability of patients and the ability to actually get a definitive answer will have to be taken into account to the benefit risk decision. One last point I just wanted to make, and it was something that Kurt said earlier that I fully agreed with, was that in that situation where you think, well, actually, do the concurrent controls look the same as the non-concurrent controls, and you feel that that's actually something that you need to lean on as part of the decision, then having additional arms in the platform that can say, yes, these all performed the same across that period as well, will help to add to that holistic benefit risk assessment. Thanks.
Olivier.
Sorry, I'm muted. Yeah, thank you. Overall, I think I agree with Andrew. And being a former regulator, I think I still err on the cautious side. I mean, I spontaneously, maybe even in phase 2, I will use concurrent control for the primary analysis, and then have as a sensitivity analysis the one using the historical information, provided that they're supporting the same estimate. I think this is where I would lie, But it's true that you have no guarantee that the two analyses, they will go in the same direction. That reminds me again, discussion around the use of historical data. I mean, I remember discussion, people telling me, well, if your historical data and the concurrent one, they're not compatible. I mean, which do you know which one is wrong? I mean, that's a difficult call. And equally, I mean, imagine you have a very significant treatment effect when you use only concurrent data. And it disappears if you provide the sensibility analysis that anyway, even if you don't provide, people will ask you to provide it by curiosity, and then you lose statistical significance. I mean, I don't think you can punish people for providing more information that they wouldn't have had otherwise, So I think that would be where I would bring the cautious. Yes, maybe there could be different results, but you could probably ask some tolerance there if that were the case, because you wouldn't see it in a normal case, right? So I think that would be my point of view here.
Thank you, Olivier. Olga.
My apologies, I couldn't unmute. So when we industry contribute to such trials, it's usually the main reasons for us, either because we cannot do those trials by ourselves, because it's difficult, maybe rare population, maybe a lot of competition for specific indication, or because of something's happening in the world, like this COVID-19 pandemic. So there is always a good reasons why we put all those resources together. If we are forced to do analysis with concurrent control, I don't think that any of us would do any sensitivity analysis. That would be enough, and we don't want to get into a situation when we have inconsistency for further discussions and further delays. If we actually allow and do as primary analysis with non-concurrent control and concurrent control, of course, the secondary analysis or sensitivity analysis should be done with concurrent control, and then the reasons if we have different results should be investigated. In my opinion, this is the most important and interesting area. We have an opportunity to learn why the results are different, what is happening with population, and maybe we can learn how to treat the population better. So again, going to my personal opinion, we should do more trials where we allow to use non-concurrent control and learn from it. But of course, as I said, I'm a statistician, I'm a scientist. We need to use appropriate methods to make sure that our inferences are scientifically sound.
And we'll have Stephen wrap up this.
Yeah, so I agree with Scott. I think that doesn't necessarily follow that when you have a sensitivity analysis, that we will always know which of the two analyses we should prefer. And there are plausible situations under which the analysis which uses more information, so going down on the minimizing variance at the risk of some increase in bias, would be the more informative analysis. And in general, once one starts looking for information elsewhere, it's not reasonable to expect that the two analyses will agree. For example, you might decide that what you're going to do is do something rather traditional and bad, which is use a responder analysis by dichotomizing patients on the basis of difference from baseline of some particular outcome. Or you could decide to use the baseline as a covariate. Both of these are actually randomized clinical trial analyses. Using the baseline as a covariate on a continuous measure might be equivalent to having run trials that were twice, 3 times, 4 times as large as the inefficient analysis. It's unreasonable under those circumstances to expect the two analyses to agree, but there we have a clear idea as to which one should be better. The problem we're facing with recovering information from non-concurrent controls is that we don't really know clearly in all the cases we would like to know how risky this is. And I'm not saying we should never do it, but I think we should make a very, very clear list of all the things that could change. And just calling them time, I think, is not good enough.
Great. With that, we will transition to final remarks. Martin?
Yes, thank you. So we have planned for a short 90-second wrap-ups, also representative sort of for the different stakeholders from academia, industry, and regulators. Stephen.
I think I've already said everything that I need to say about this. It's been an interesting discussion. I don't know to what extent the individual participants have changed their positions. That in itself shows how much prior prejudice is important when one comes to evaluate results.
Thanks, Olivier.
Yeah, thank you. I mean, again, not sure there is a very specific industry view on that, but I really enjoy it. It's clear that there's a trade-off, again, a benefit risk to have in mind. I mean, we have different point of views there. People that are a bit more conservative, others that are more daring, but I think people are very clear in mind that this is mainly driven by type 1 error control and bias. Maybe one thing that I would like to emphasize, for me, the clear winner of today is transparency. I really enjoyed the 2 first presentations. That was really refreshing to see the math and the different models and what do you gain, what do you lose, and under which assumption. I think that was very new. on the topic as compared to what had already maybe said and written. So that's a clear win for me today, that aspect. That will really help in the discussions, specifically with regulators. So thank you for that.
Thanks. And then, Daniel?
Thank you. So first, I want to thank the organizers for putting this webinar together, as well as the perspectives from my fellow regulators and the representatives from academia, industry, and payers. So on the regulatory side, I do think you heard a fairly unified perspective from Andrew and Julie and myself that in many situations, probably the first tool we would be looking for would be the concurrently randomized comparison due to the issues that have been brought up throughout the day related to comparability. I guess the final point I wanted to make is that I think it's a very important point that came up in the discussion, that a lot of the topics today have centered around the efficiency gains from non-concurrent controls, but there definitely are efficiency gains in platform trials, even with the fully randomized comparison related to the shared infrastructure and the ability to share control patients, even with the fully randomized comparison. So I think it's important not to lose sight of that. Thank you.
Yes, thanks a lot. So it's left to me also to thank all the excellent speakers and the panelists for the talks and lively, open discussion. I think sort of a recurring topic was the variance bias trade-off, and I think Stephen showed this quite clearly, sort of also sort of the different settings where one would put more emphasis on the bias and others more emphasis on the variance. It's probably also from the perspective of different stakeholders that some may be more concerned with bias, some more with the variance. But I think I agree with Olivier, sort of where the perspective does not really matter is when it comes to the agreement on the facts, namely what the properties of the different methods are, and it's our role as statisticians to be really transparent and transparently communicate the underlying assumptions, benefit, and risks, which is then the basis for a sound and interdisciplinary decision-making on the choice of the control groups for all the stakeholders. We also saw that there is no one-size-fits-all approach, and the best strategy will depend on many factors. And I'm confident also that this meeting is not the end of the discussion. There's still a lot to be learned and also developed with regard to methodology and especially experience with regard to the use of these methods. Next slide. So the EU Pearl project has still a year to go, and the work on non-concurrent controls is only one part of the work. There's a lot of other work streams on statistical, regulatory, operational, and legal aspects of platform trials. Let me mention the large stakeholder workshop that is planned for later this year in autumn. You'll find more details on the website of EU Pearl, where you can also find publications and a newsletter. Next slide. Here you can also find details on how to stay in touch with EU Pearl. With this, let me thank my co-moderator, Peter Masonbrink, and conclude the session. Many thanks for your attendance.
Thank you all.